Do you think it will go away again once you pull up your PowerPoint, though? Or do you think
Let's see. So we're so now I'm going to start my screen share. And how is that? So can you see that?
Yeah. Everything looks perfect on my end, but I just still can't figure out the record button.
Okay, so my little, pulsing red dot is showing record. So we will get started.
Okay, perfect. Yeah, and then we'll cut us talking out of it. So yeah, you're good. You can go right ahead and I'll send you a Dropbox link to upload the video when you're done.
Okay, super.
All right, thanks so much.
Hi, everyone. Thanks for having me at this first virtual TACA meeting. I'll be talking to you today about mitochondrial autism and the cell danger response, and give you a little update on our Suramin work. So the disclosures that are most important include this one in blue at the bottom that says that there's no approved use of Suramin in the United States, and it's illegal to import Suramin for human use without explicit FDA and IRB approval and an IND. So here are some references and resources. The PMID numbers allow you to just copy those and go directly to PubMed to look at any of our papers on the cell danger response, on the environmental components of human health, metabolic features of the healing cycle, and many other studies. So the big question, number one, is, are the behavioral symptoms of autism caused by a treatable metabolic syndrome? So our work is really focused on trying to answer this question. So for the first 100 years of autism research, we spent a lot of time looking at the different triggers, the things that could increase the risk of autism that include early life stress, genes, air pollution, flame retardants, metals and pesticides, infections, fever, various cell dysfunctions and cytokines in the microbiome. But each of these can be thought of as a different way to ring the same cellular bell. And so when Grunya Shukareva first published her 6 children with ASD in 1925, and then Leo Kanner independently published his work in 1943, they're really focusing on the clinical characterization of autism spectrum disorder. And now what I'd like to do is show you that all these triggers can produce a unified cellular response that we call the cell danger response that can lead to ASD and myalgic encephalomyelitis, chronic fatigue syndrome, and many other disorders. So we're kind of transitioning, reframing the question from the diversity of triggers that can cause autism now to the unity of the cellular response. Okay. So extracellular ATP is a key regulator of the cell danger response. Here's a cartoon of a cell that emphasizes this point that whenever a cell is stressed, it leaks the ATP that's made inside to outside. And in that way, it's kind of similar to a water balloon under stress, or if you can imagine 1,000 little pinholes. When that water balloon is stressed, there's more of those cellular contents that are released. And in particular, they're released in accordance to the mitochondrial sensing of cellular changes. So mitochondria sense those changes and they send signals to the nucleus, and the nucleus talks to mitochondria. And then we have ATP being released through specific channels in the cell membrane. And there are really many, many of these different channels. But you can imagine when they open up, the ATP that is necessary inside the cell to help a cell grow and repair and function now gets lost to the outside. We call that a dissipative ATP loss. So I went looking for any molecule in the world. None of these were known at the time, but any molecule in the world that could specifically act like by inhibiting the efflux, the loss of the ATP from those channels, and also be able to work on the cellular receptors that are so-called purinergic receptors that go by names of P2X and P2Y receptors that send messages to the nucleus that change cell function, okay? And so when we did that, we found one drug, and that was Suramin. Now, the point I'm going to emphasize in the talk today is that the cell danger response is universal to all life on Earth. And that is specifically if the cells of those organisms have mitochondria, okay? And that's universal from plankton to people and plants. And here is a little mustard plant that has been bitten over on the right-hand side of your screen by a cricket. So that's a little bit of damage in the leaf. This plant's been modified so that it will fluoresce when calcium is released from its cells. So it turns out that if you add either glutamate as a danger signal or ATP as a danger signal, the following thing happens. The message of the cellular damage or danger is propagated throughout the entire plant within about 90 seconds. I'll show you that one more time. Okay, obviously, with a nervous system, this can be done even more efficiently. Okay, so next. So ATP is also a modulating co-neurotransmitter at every synapse. So here's an astrocyte. This is a presynaptic membrane and a postsynaptic membrane. And glutamate, in this case, is being released. So again, just like the previous slide, glutamate is a kind of signaling molecule. But in addition to glutamate, ATP is also co-released and modifies how the postsynaptic membrane responds to neurotransmitters. So how can we put this together in this context of life? So under normal circumstances, we spend each day waking up and being active, and we take in nutrients. And as part of this, the normal activities of the day, our cells will release a little bit of ATP. Okay, so the extracellular ATP is increased, and the CP1 is a checkpoint 1. But that ATP is easily metabolized into things like adenosine, which are actually necessary to induce sleep. So with sleep, it's further metabolized, and we return back to wakeful activity and nutrient intake. Now, if there's been severe injury that might come from a bacterial infection, a viral infection, a broken leg or a heart attack, what happens is we pass into this first phase of the cell danger response, what we call CDR1, which is the one that is designed to contain that injury, okay? And that involves innate immunity and inflammation. And really, any increase in the kind of stress, whether it's chemical, physical, microbial, can produce this. And this is off to the right is a video micrograph of mitochondria in how they literally change their structure within 5 minutes of a cell that has been placed under stress. And so I'll activate this and show how mitochondria go from this long filamentous configuration to a more spherical or fragmented configuration. And that's called the spaghetti to meatball transformation, but also specifically involves the movement, the change in mitochondria that are filamentous, in the so-called M2 mitochondria, to this pro-inflammatory M1 mitochondria. Okay. Each one of these, facilitates different biochemical characteristics for the cell. But the main point is that in order to heal, we actually have to move through 3 different stages of mitochondrial function in order to reset and get back to normal. And we can have blocks at different points in this cycle that are called, that we just talk about checkpoint 1, checkpoint 2, checkpoint 3. And that we believe that a large portion of autism and chronic fatigue syndrome result from blocks in this checkpoint 3, so that there's a repeating loop in this stage, in this last stage, that does not allow cells to get back to normal health. Okay, so what's another way of looking at this? We can look at the single gene causes of autism, and it turns out, interestingly, they also show the central importance of the cell danger response, whether you're talking about the Fragile X gene that can be, when you inactivate this gene, it since the protein normally will bind to certain mRNAs that are pro-inflammatory. If you're no longer binding and inhibiting those pro-inflammatory cytokines like TNF-alpha, autism symptoms can be produced. And same thing for the Rett syndrome gene, the MECP2. It alters the chromatin configuration and starts to open chromatin and results in the activation or mobilization of endogenous retrovirus elements and endogenous mobile elements, including this L1. Angelman is due to a defect that will hit this ubiquitination, this ubiquitin ligase protein, and that prevents the cell from normally ubiquitinating unfolded proteins and results in, again, all disruption of the normal cell danger response, which depends on the ability to sense, unfolded proteins. Smith-Magenis is a defect that leads principally to the abnormalities in this retinoic acid-induced gene that's important for the antiviral response. And Smith-Lemli-Opitz is a defect in the 7-dehydrocholesterol reductase gene that results in decreased amounts of cholesterol, and because you need cholesterol in order to make cortisol, which is a central stress hormone, the inability to mobilize the normal stress response also activates the cell danger response. So when we look for anticholinergic drugs, it turns out they don't affect the central, the core features of the actual gene function, okay, which lead to these recognizable syndromes. They actually just affect the behavioral symptoms that we associate with autism. Okay, so what about a quick historical recap? So this is just to let you know that this is not something that just popped out of the blue last year. This is something that is work that has been growing in momentum since the year 2000. So in 2000, we identified the first mitochondrial DNA mutations that caused autism, and found that in contrast to the simplest interpretation, which would be that mitochondrial defects cause autism, that turns out not to be true. It turns out that it's an imbalance in the energy produced by mitochondria, and that some aspects of mitochondrial function were actually increased 250%. Okay, so this is what we call a segmental overfunction problem that was produced by the mutation. And you might think of this as an imbalance in the force, so to speak, that mitochondria generate. We took 5 years to understand how mitochondria regulate healing, and that resulted ultimately in the birth of the cell danger response concept, and to this idea that potentially the CDR, the cell danger response, was acting like a plate glass window that prevented children from, breaking out into the world and engaging in normally in normal child play. So there are a number of other events that have all contributed to where we are now, including an NIH conference on mitochondria and autism that I organized in 2010, all the way down to showing that Suramin worked in an environmental mouse model of ASD. It even worked in adult mice with ASD symptoms, and it worked in Fragile X model of ASD in mice. Okay, so that was 2015. We then did a clinical trial that I'll talk about at the end. But in the meantime, there were a lot of other people that began to pick up on our work. And so here's an example that international studies of children with ASD also confirm the central importance of purines and mitochondria. So in this study by Persico and his colleagues, purines were the most significantly disturbed metabolic pathway. And the other metabolic pathways all trace to mitochondria as well. In a paper that came out last year, it was found by Horvath et al. That just the injection of ATP itself during pregnancy into a pregnant female reproduced all the features of the maternal immune activation mouse model, this classical model of AST. And so you didn't have to inject double-strand RNA, which is the classical way. You could just inject ATP and produce all of the social abnormalities and even the neurological abnormalities that are most graphically illustrated by a dropout in cerebellar Purkinje cells that happen. So these are Purkinje cells, these brightly fluorescent green cells here in the cerebellum. And then animals that have been treated with double-strand RNA, the poly-IC, have a dropout. But also there's a dropout in Purkinje cells after ATP injection. And the behaviors were indistinguishable from the behaviors of the classical MIA mouse. In addition, when the authors used antipurinergic drugs, they could normalize the behavior of these animals. Okay, so it turns out that there's a rat model where a pregnant female is given valproic acid and her pups will show social abnormalities and learning disabilities for life. But it turns out that, so that's illustrated by this time spent sniffing, which is the social interaction time of animals, either with another rat or a novel object. And it turns out if you treat those animals with Suramin, that you get complete normalization of the social interaction with rats and a preference over inanimate objects. And finally, just this year came out a study that was published in Cell that showed that even the Fragile X model of ASD had a root problem with ATP leak. So this is a picture of the ATP synthase in mitochondria that makes ATP, and that MATP is mitochondrial ATP. But it leaks out of the mitochondria to increase cellular levels of ATP that then can leak out of the cell membrane to produce extracellular ATP that then activates the cell danger response and inflammation. Yeah. So a lot of convergent, independent work on this concept of extracellular ATP being a final common denominator in many, many different causes of autism. Okay, so what happens if you direct treatment to the cell danger response in children with autism? And so we did this small clinical trial of 10 children that was published in 2017 that was called the SAT-1 or Suramin Autism Treatment 1 trial. And so this is an illustration of mitochondria, and I'm going to just illustrate, show a cartoon that discusses our results. So we wanted to look at whether Suramin could actually treat autism. And specifically, when you illustrate this with a picture of a neuron with a nucleus and mitochondria, when that neuron is stressed, ATP can be lost from the cell and then bind to receptors that then trigger the cell danger response. So if you then treat these cells with Suramin, illustrated by the little S's, they not only block the loss of ATP from the cell, but they also prevent the activation of the cell danger response, help to normalize it, and allow the neurons to redirect resources and energy toward healing and communication. So the overall design of the study was that the children were paired by age, IQ, and ASD severity. It was prospectively randomized and double-blind, placebo-controlled, 10 boys, 5 to 14 years of age, given a single dose of Suramin, and then tested before and after with a classical neurodevelopmental test called ADOS2. So these are the pictures of the 10 pioneers, the children. And we had 4 boys who were actually nonverbal and with severe oral motor dyspraxia. But within starting to give, give infusions, which we did not know at the time were either Suramin or saline, what happened is half the boys started having dramatic improvements in language and social interaction, and the other half had no change. And when we broke the blind, we find that the half that had improved had received Suramin, and the half that did not had received the placebo. So when you analyze the specific quantitative results, you see that ADOS scores were dramatically improved with p-values you don't normally achieve unless you have 20 or 30 or more children of 0.0028. But the placebo group had insignificant changes, no changes at all. And then the aberrant behavior checklist and ATAC and clinical clinical global impression scales also showed significant improvement, but there were no improvement in those scores in the children who had received placebo. So to summarize, the average child in the study who had received Suramin started out with a score of about 8.6 on the ADOS scale. And remember that from 7 to 10 is on the comparison score for ADOS is by definition autism spectrum disorder. So below that technically does not meet ADOS requirements for autism diagnosis, and above it does. So after 1 dose, the children who had received Suramin had dropped to an average score of 7 with very significant changes, and the placebo group was completely within the error bars after their single dose. And so it raised the question, if 1 dose produces this amount of change, if you give a dose per month for 3 months, will some children come off spectrum? So the results can be summarized as follows. A single low dose was safe. Each of the 5 children who received it showed improvements in language, social interaction, and decreased stims and restrictive interests. But in addition to the core symptoms, there were also non-core symptom improvements. So accelerated development and learning milestones at school noted by their teachers, who didn't actually know that the children were part of a clinical trial. Decreased anxiety and meltdowns in novel situations, improved sleep and new interest in foods, and improved GI function. And I can say from our mouse work, we know that Suramin restored normal microbiome diversity within a month. And that there was magnified benefit from all the usual ABA speech and occupational therapies that for some children had remained stagnant for months. So, but none of the 5 children who received placebo showed any of these improvements. Okay, so the next big questions are, is Suramin safe and effective for the treatment of all kinds of autism in both boys and girls? And can Suramin help some children to even come off the ASD spectrum? We don't know the answer to that, but that's what the next trials will be designed to answer. So coming in 2021, there'll be the Suramin Autism Treatment 2 study. That will be sponsored by Kuzani Pharmaceuticals to test Suramin autism. And the plan will be to test Suramin in 60 to 80 children with ASD. And just a note, Kuzani is a new American development stage company that is making a new preparation of Suramin for the next clinical trials. We also have another project that I'm going to close with called the Newborn Screening Project that's designed, that makes use of a new test that we've developed that will look at over 2,000 metabolites and environmental chemicals in dried blood spots that were collected at the time of birth in children. That means that they were stored to look for genetic disorders long ago, are available to look at different chemicals that were not measured originally. Okay, so this is where I want to ask for your help. So I need your help for this new study. We are planning to enroll 400 families, that's 200 with ASD and 200 controls, ages 3 to 10 years old. And this is currently restricted to California, but it will be extended to other states soon. And if you want to learn more, look at our website. Please note that there are no new blood samples required. Anybody who participates, will receive a $40 Amazon e-gift card. And all we need from you is 15 minutes of your time to fill out an online form and give us your permission. And you can help make this study possible. So I'll tell you a little bit of an update. This study has been open for a year. We have been actively recruiting, but the enrollment has been slow. We've only got about 170 families out of what we had hoped would be 400. And so please let people know, and please look at our website. This really could help many, many, many children in the future. So what do we do in our lab? Treatment and early diagnosis for optimal outcomes. So these are really the two things that we're focusing on. And you've heard about both of them today, both about Suramin for the treatment of autism and this root problem of persistent activation of the cell danger response, and then also about newborn screening. So what's the human impact? We think it's priceless. And I thank you very much for all your help. And here's a list of some of the people and foundations that have supported all the work today. So I'll end it here and say thank you very much, and have a wonderful rest of the meeting. Okay, bye-bye.